Hello. If I could just have you fill this out, just by the end of the session, not now. Yeah. We good? Cool. Here's this and this. That's just to fill out by the end of the session. All right, welcome everyone. How's it going? Ready for the final? Yeah, kind of? Yeah. Before we begin, make sure you fill this out by the end of the session. This is just a feedback form. You'll just turn it back to me. Just make sure you fill out the this bubble, the bubble in the number 37, and then put in your UCF ID. That's just to make sure that you're actually a student that's taking the class and not someone that hates me and just comes here to give me like a really, really bad review or something like that. So, yeah. Is there any specific questions as far as material that you'd like me to go over? Things that you're not super sure about? So the final is 100 questions, right? Remember that this final will replace your 5th test, basically, and it's going to replace the 2nd lowest grade, correct? Yes? So you get double the opportunity here to improve your grade. So make sure you study. As far as knowing exactly what to study, it's a little bit hard because it is a lot of material, as you know. But there are many things that are repeated throughout the semester. I don't know if has anybody noticed that by now. I hope so, right? So it can be very specific things, right? Such as, I don't know, the first thing that always pops in my mind that's very specific that gets repeated is transcription factors, NF-kappa B. How many times do you see NF-kappa B, for example? I know this is out of nowhere, but does anybody remember when, like, in which cells you see NF-kappa B? What's that? Macrophages, cool, awesome, great. With macrophages, it helped to produce cytokines, inflammatory cytokines, so that we can just have inflammation whenever there's a bacterial infection, for example. And it's the whole cascade of protein that you study with the toll-like receptor, right? With the CD14, MBP, MID88, blah, 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 blah. Another place where you see it is, for example, with B cells. If you remember, CD40 ligand interaction allowed NF-kappa B activation, which did a bunch of stuff. One of them was ICAM transcription, and the second thing was BCL-XL production, for example. So try to look for those things that you see back and forth. That you see over and over, that you've seen over and over throughout the semester, and you know it's a better chance that it might show up on the exam, right? Those of you who haven't got in here yet, that just came in, make sure you grab one of these by the time you leave. I'll just pass it around, and whoever didn't grab one, they're just feedback forms. So yeah. Just make sure you do that. Go over the practice tests that I have. The practice tests might be a little bit too specific because I did them thinking about the exam itself and not the final. So some questions might be a little bit too specific for the final, but if you can remember those little details, it might help you out when you're taking the final, and it might be that, you know, the difference between an A and a B, for example. Do the study guides if you can. Just do them all over again. Just try to start answering those questions that he has on each study guide. And whatever you don't know, just go back and review it, right? And even if you answer it, just go back and review the PowerPoints and make sure that there's nothing else that is important that you missed. And, oh, go see your tests. How many of you have seen previous tests? Okay, is he giving a chance to see the tests? Today was the last day. Ah, that sucks. Okay. Well, those of you who went, good job. Because I think seeing the test is one of the biggest things that you can do to help yourself. Because a lot of the times it's not that you don't know the material, is that you maybe misread something, you maybe skipped a word, or whatever it is that made you lose that point, right? So if you were able to, then good for you. Where are the evals? Over there. Can you just pass them down for me? If you just came in, just make sure you grab one of those evals and do them before you leave. Bubble in that number 37. So yeah, if nobody has any specific questions, what I'm going to do is I'm going to go through the test. I wrote down a few things that are important to mention for each question that might be helpful for you to kind of review everything. And if you have any questions at all, just stop me and I'll answer it. I don't care. I rather you ask me questions than me just ramble on for 2 hours and not know whether I'm helping you out or not. Okay? Yes? Okay, cool. So, oh, by the way, the answer key, I have it here, but I'll be uploading this as soon as I get out of here. I have my laptop and I'll be uploading it. Okay? Cool. So, question. Oh, if I start writing and you can't see because I'm writing outside of the screen, just let me know and I'll move as well. So, which of the following is not? Yeah. Yeah. All right. So, which of the following is not characteristic of inflammation? Inactivation of macrophages, increased vascular permeability, vasodilation, pain, and influx of leukocytes. Which one is not part of inflammation? A, right? So you need activation of macrophages because they're one of the ones that will cause the inflammation. Correct? Any questions? No? All right. Which of the following lymphocytes has an orange, red, stained granule and is specialized in fighting parasitic infections? So which lymphocyte has red, orange granules and fights parasitic infections? No, it's not a lymphocyte. He has that question in every test. Come on. Doesn't he? Or almost. So make sure when you're reading, so when you're taking this test, it's 100 questions, right? Make sure you underline all those key words, okay? So if it's a lymphocyte, what cells fall under the lymphocyte category? T, B, and NK, right? So lymphocytes, it's going to be T, B, and NK. Now, leukocytes are going to be Ben, monocyte, macrophages, dendritic cells, and so on, right? So just make sure that you can differentiate that and you can figure out those keywords. So if the question asked which of the following leukocytes has orange, red, stained granules, and so on, then the correct answer would have been eosinophils, right? Does it make sense? Yes. Cool. All right. Pyogenic refers to the body's production of heat to fight infections. True or false? What does it mean? Pus, right? So pyo equals pus. The way I remember it is I say to myself, yo, that's pus, right? So it's pyo, pus. I know it's kind of silly, but it kind of stuck with me. So yeah, so 3 is false. So complement. Do you think you need to know complement for this test? Yes. Would you like me to go over complement? Like the alternative and classical really quick? Raise your hand for yes. Raise your hand for no. Okay, cool. I'll make sure I upload a little worksheet that I have. It's really easy, and you can also, there's a couple of videos that are pretty good as well that you can look. But I'll get through this. If we have enough time, then I'll go back and go over it, okay? Yeah. So just to answer these, which protein initiates the classical pathway? Is it C1q, C1s? C3b is not going to be it because that's going to be the alternative, right? So we're going to take this out. Is it C1q or C1s? Why is it not C1q? What's that? So close. What's that? Yeah, C1q is the binding domain of the protein C1. So C1 is made out of 3 domains, C1q, R, and S. C1q binds to 1 AGM, 2 IgGs, or 1 CRP, C-reactive protein, remember? And when it binds to the 1 IgM, 2 IgGs, or 1 CRP, it will activate C1R, and then C1R activates C1s, and C1s is the one that has that catalytic activity that will allow you to start cleaving what? C3, C2, or C4? C4 and C2, and then that's going to produce what? C4b2a, which is the C3 convertase of classical pathway, great. That is going to cleave C3b, right, into C3a and b. The C3b can either land somewhere nearby and opsonize. It can bind to that C4b2a itself and form the C4b2a3b, which is the C5 convertase of the classical pathway, which will then form the MAC, right? The membrane attack complex, the pore that will kill the bacteria. Now, that C3b can also turn into what? Going back a step. A C3 convertase, can it turn into that? Yes? So C3 convertase that itself can turn into a C5 convertase as well. Okay? So when you're reviewing it, just when you draw it, make sure you try to connect how both pathways are related. Okay? Does the lectin pathway have like different complement proteins? It's going to be what? It's going to use which ones? Yeah, it's going to be the same as a classical pretty much, C4 and C2, and then it ends up feeding into the alternative. Cool. Good job. So which one's the receptor for C3b? Oh, the answer for this one is C1s. Thank you. So again, the C1 is the protein that has 3 domains, 3 subunits, C1q, R, and S. Q binds to the bacteria, well, it binds to 1 IgM, 2 IgGs, or 1 CRP. Once it does that, it activates C1R, and then once C1R is activated, it will activate C1s. And then C1s is the one that has the catalytic activity. What's that? By the word initiate. No, because it doesn't do any cleavage. Yeah, you're talking about the beginning of the cascade, and the beginning of the cascade is when you start cutting the first product. Yeah, so that's why it's C1s. What's up?
If C1R was an option, would that be correct?
No. No? Because C1R is like in between Q and S. So it's C1Q activates C1R, which activates C1S, and then C1S will form the C4b2a. Make sense? Everyone? Yeah? Okay, cool. So what's the receptor for C3b? Anyone? CR2? CR1. What does CR2 bind to? IC3D, right? We saw that on this last task for B cells. B cell activation, remember? What about CR3? IC3B. So C3D, IC3B. And C3B and IC3B, those are both degradation products of the complement cascade, and they're made so that you can continue to opsonize stuff, but you don't have more complement activation. Remember, you had the factor H and I. So C3b gets cut by factor H and H and I, sorry, and it will turn into IC3b, B as in boy, and then again factor I will come in once again, and we'll cut IC3b into C3d. Okay? Yeah? Cool. So going over the toll-like receptor, what extracellular proteins will the toll-like receptor need to be able to start the cytokine-producing protein cascade? So what is needed besides the TLR outside the macrophage to be able to start the toll-like receptor cascade? Yeah, so that you can produce cytokines. Anyone? Yeah. All of the above, yeah. So you're going to need MD2, CD14, and LBP. MD2, so LBP and CD14 are involved with holding to the LPS, basically, and MD2 is associated with the toll-like receptor. If you don't remember this, you can go, did I write the 6? No. You can go to chapter, I want to say 2, and there's this one picture that has the whole cascade, and it has drawn all the proteins that you have on the toll-like receptor, just so that you can refresh your mind and remember what happens in that instance. All right? So number 7 is asking to identify the mismatched pair. ICAM with LFA1. Do you think they go together? What is an ICAM and what is an LFA1? If you'd have to give me like the, what kind of family of protein do they belong? You can be very broad. You don't have to tell me specifically. What's that? So they're adhesion molecules. They're not selectin vascular addressins. They're integrins and immunoglobulin-like superfamily. Yeah. So these are good together because they interact with each other. CD14 and LPS, they also interact with each other because they're part of the toll-like receptor. NK cells and interferon gamma, are those? Is it okay if they're together? What was interferon gamma? This goes back to like chapter 2 again, I think. Yeah, it activates macrophages. It's secreted by which type of cell? NK cells, right? So it's secreted by NK cells. It activates macrophages, among other things. So yeah, they're good together. This is also part of like that chapter. It's either 2 or 3. If you don't remember it at all, I'd recommend you to go over and review it. And then remember, just going off of the interferons, you also had interferon alpha and beta, which were secreted by all nucleated cells, and they just kind of helped with fighting viral infection, making the environment inside the cell a little bit harsher for viruses, as well as letting the nearby cells know that, hey, I'm infected with a virus, get ready, basically. Yeah? No, that's different. Yeah, so interferon alpha and beta, they kind of activate themselves. So it's an autocrine type of signaling, and they basically activate molecular mechanisms in the cell so that it's harder for viruses to continue to replicate in there. Yeah, and then they do this, they signal nearby cells as well. And then selectin with tight binding, this is wrong because selectin is involved in rolling adhesion, if you remember, with the whole neutrophil homing. Okay, cool. So catalase is the enzyme used by neutrophils to produce hydrogen peroxide. Is that true or false? If you took micro, you should know this. It's false. Yeah. Catalase breaks down hydrogen peroxide into water and oxygen. Yeah, thank you. That's why if you ever had like a little scrape or a cut and you spray some hydrogen peroxide, that's why you get all the bubbling, because there's catalase in there that breaks down the hydrogen peroxide. All right. So interferon alpha and interferon beta are produced by which of the following cells? Only NK cells, only T cells, all nucleated, A and B, or none of the above? I just talked about this. All nucleated, right? Cool. Awesome. So we're talking about antibodies now. So one binding site of any antibody is composed by how many hypervariable regions? So before we answer this, what is a hypervariable region? Who can tell me really quick what that is? What's that? Yeah, what forms the binding site. Why is that important to have hypervariable regions in the binding site of an antibody? What's that? It gives us affinity. I'll give you that. How about it's because they're very, very variable? That's why they're called hypervariable, right? And it allows us to bind to many different types of things, peptides, viruses, bacteria, whatever it is. Because remember, your hypervariable regions were coded by your V, D, and J segments, right, on your antibody. So basically, if you had to draw an antibody, your hypervariable regions were going to be right here, right? So if you had to zoom in into this, this is a very bad drawing, I'm sorry. But you basically have HV1, HV2, and HV3. Oh my God. Let me draw this again. So you have your antibody. So that's basically it, right? If you were to zoom in here, your hypervariable regions would be composed, 3 of them would make up the binding site, half of a binding site basically, because you have 3 for your light chain and 3 for your heavy chain. Do you all remember that? Yes? They were coded by your V, D, and J and V and J. V, D, and J is going to be for which chain on the BCR? Heavy chain. V and J is for your light chain. Now, if we're talking about TCRs, will TCRs have hypervariable regions as well? Yes. Will they have VD and J and VJ segments? Yes. Okay, cool. And they're the same thing. They're created the same way through somatic recombination with RAG1 and 2 and TDT and all that. It's just, you know, you're dealing with either genes for a BCR or genes for a TCR, right? But the mechanisms are very similar, if not the same. Okay? So if we have 3 for, let's say this is the light chain, and then the heavy. What's that? I didn't hear you. Oh yeah, that's a good question. What's the other name for hypervariable regions? CDRs, right? Complementary determining regions. I don't think you need to know that. So you have 3 for the light chain, 3 for the heavy chain, so that's a total of 6. Cool. Now if we're talking about how many hypervariable regions are you going to have in dimeric IgA total? 24, right? Because IgA, dimeric IgA is going to be 2 IgAs together, right? So in each you're going to have 12 because it's 6 and 6. So 12 plus 12, 24. IgM is going to have, IgM is pentameric, right? So 60, right? Make sense? Yeah? Cool. All right, so a little bit more about your complementary determining regions. Can you see both here? No. So question 11 is asking about the third hypervariable region of the light chain is determined by which segments? Who remembers that? What's that? It's the V and J junction, right? So as a rule of thumb, for the light chain, your CDRs 1 and 2, they're going to be coded by your V segments, V and J segments. But the CDR3 is the most variable, and that's because it's going to be coded by the junction between the segments. That's for the light chain. For the heavy chain is the same thing. The CDRs 1 and 2 from V segments, and then CDR3 is going to be coded by the V and D junction, D segment, and D and J junction. Does it make sense? Because your heavy chain, so if we had to draw your DNA, for your heavy chain, you're going to have let's say this is going to be DNA that's been through somatic recombination already. So it's going to have 1V, 1D, 1J, and the constant region, right, IgM. So you're going to have 1V, 1D, 1J, and then your constant M. So with the DNA for your BCR for the heavy chain is going to have first your VD and J, then your IgM basically gene, then your IgD, right? So what happens is that the CDRs, so HV1, HV2, so the CDRs 1 and 2, they're going to be coded by the V region, while the CDR3 is going to be coded by the junction between V and D, D, and then the junction between D and J. Because remember, the junction is when you add those nucleotides. When you were sticking the pieces together of DNA, you had TDT that added just random nucleotides in there so that you could have one DNA strand instead of having pieces of DNA floating around. Does it make sense? Yeah? And then the same thing for the light chain is just you don't have a D segment, so it's just going to be V and J and the junction, pretty much. What's up?
So what you're saying is that it uses the junction.
Speak up a little bit, because
Uses the VD junction, then the VJ junction, is that what you're saying?
yeah, that's what gives the diversity to that CDR3 region. And that's why if you were to grab a sample of your blood and start typing your different CDR, your antibodies and compare your CDRs, the third region of the CDRs, that's going to be the most variable one, pretty much. Okay? if it hasn't sunk in yet, just, let it go for a minute, and then tonight when you're reviewing stuff, just think about it again, and it'll all make sense. Also, you can look at the recording online on YouTube, because they're filming right there. Hi, Mom. So you can just see everything again, and you'll be able to see that too online. So, all right? Yeah? Cool. So to answer this question, the third hypervariable region is going to be determined by your VJ junction. This is in the light chain, right? If it was asking the heavy chain, it was going to be the VD, D, and DJ junction. So now it's asking about secreted versus membrane bound. So that decision, instead of being made in your DNA, it's made in your RNA, right? And just as a quick refresher, remember that, so first of all, remember you have DNA, RNA, protein, right? This is the dogma of biology. Everybody should know this by heart. What happens is that here, this is your DNA, right? So we're at this point right here. But then this gets transcribed into RNA, which is, it'll still have your VD and J, your constant mu, your constant delta. But then it's also going to have two extra regions that is going to determine whether it's secreted or membrane bound, right? So basically what your cell does when it's going through somatic recombination and maturation in general, if it needs to put BCRs on a surface, with the RNA, it's going to do RNA splicing, and it's going to cut out this section right here, the secreted one, and just leave the membrane bound with the information to create the IgM. If it needs to secrete IgM, it's going to cut out the membrane bound and leave the secreted. And then it also has the option of choosing in the RNA whether you can make IgM or make IgD, because you have your C mu and C delta genes over here. So you cut out whatever you don't need, right? And you just produce, you make that RNA into a protein. Does it make sense? Yeah? Okay. Cool. So that's going to be RNA splicing of secreted versus membrane bound. Now, as far as somatic hypermutation, are we going to have reg 1 and 2 on? Hypermutation. Are we going to have TDT on? Oh, it's not an option, but are we going to have AID on? Yes, right? So AID is going to be what changes a C to a U, and then that U is going to be changed to whatever DNTP is available right there, and it's going to make those point mutations, which will either increase the affinity of your antibody, decrease it, or just leave it the same. And that happens in the bone marrow or in the lymph node, somatic hypermutation. Somatic hypermutation happens in the lymph node or in the bone marrow? Somatic hypermutation happens in the lymph node or in the bone marrow? In the lymph node, in the follicle. Now, somatic recombination will happen in the bone marrow or the thymus if we're talking about T cells, right? Does that make sense? Did you have a question?
Yes. Now, for A, it changes the C to a U. What exactly does that mean?
So you change like your nucleotide, the C, to a uracil, and then that uracil, because it's not supposed to be in your DNA, you change it again to something else, random. And that's where you make your point mutation. Yeah, just know that if you're, you can always go back and review the PowerPoints really quick, but just knowing that is enough for you to if you were to be asked a question about that, you can get the question right pretty much. Okay, yeah. So the next question is asking about your TCR and your T cell maturation. So remember that your T cell maturation is similar to your B cell maturation, right? You're going to make your TCR first, just like the B cell does its BCR. The T cell is going to make the beta chain first, test it, and then if all works well, it's going to move on and do your alpha chain, right? Just like the B cell did the heavy chain first, it tests the heavy chain, and then it goes to the light chain, basically. So basically, this question is asking you, what are the, well, I'll just read it. Once a T cell has rearranged a TCR, which of the following membrane proteins will the TCR need to leave the endoplasmic reticulum to the final destination, which is the surface of the cell? So what do you need in a T cell next to the TCR for the TCR to be able to be secreted? The CD3 complex and your zeta chains. Now, if we were talking about your BCRs, it's going to be what? Yeah, Ig alpha and beta, right? And the same thing goes for when you're talking about that checkpoint in between in the maturation. Your T cell is going to make that pre-TCR, right, to check whether the beta chain is working. And it has to go to the surface, and for it to go to the surface, it's going to need CD3 complex and zeta chains. The B cell maturation process is the same thing. You'll need your pre-BCR, right, with your lambda 5. You remember that? No, that's fine. I'll get to it. But your pre-BCR receptor, which is just the beta chain with a surrogate light chain, it's also going to need the Ig alpha and beta on the side. Okay? So in T cells, the CD3 complex is made out of 2 epsilons, 1 gamma, 1 delta, 2 epsilons, 1 gamma, 1 delta, and 2 zeta chains. Okay? Now, if it were to talk about B cells co-receptor is Ig alpha and Ig beta. Okay? As you're studying for this, for the final, because it's so much material, it's very useful to compare B and T cells. Because that's how you're technically studying for, how the book lays it out, is that it's always comparing B and T cells. So it's always good whenever you can to compare them, right? In this case, you can because they both have co-receptors, they're just different. So you can talk about the co-receptor in the T cell first, and then the co-receptor on the B cell. Question? Yes, but it's your complement receptor, right? The actual co-receptors that your BCR will always need to be able to, function and send the signal inside it are going to be Ig alpha and beta. Yeah, good question though. So, always talking about your TCRs, it's asking about the importance of the zeta chain, which is going to be what? Your zeta chain helps sending the signal to the inside, right? So signal transducing. So when your TCR binds to something that it recognizes, which is going to be an MHC molecule with a peptide, the TCR itself cannot send that signal to the inside of the T cell to activate the cell and induce proliferation and all that. And that's when your CD3 complex and zeta chain comes in, where you're going to have the whole phosphorylation with ZAP70, PIP being cleaved into IP3 and DAG. Do you remember that? Yes? Okay. So just to answer this question, the importance of the zeta chain is that it will be fundamental for that signal, to send that signal from the outside to the inside, basically. All right? Cool. 16. MHC1. Does anybody remember? Do you all remember the structure of MHC1? You had. Yeah, so you have alpha 1, alpha 2, alpha 3, and your beta 2, microglobulin, right? So the question is asking about the structure. So it has 2 transmembrane chains with 2 alpha chains and 2 beta chains. Is that true? No, right? I just drew it. It's not right. So this is wrong. It has 1 transmembrane chain, 3 alpha chains, and 1 beta 2 microglobulin. Is that correct? Yeah. Has 1 alpha 1, 1 alpha 2 that will form the peptide bonding site. Alpha 1 and 2. So that's true. Beta 2 microglobulin will also have a transmembrane chain. That's false. So then the correct answer is going to be E. Right? Knowing structures, it's always going to help you out a lot, to be honest. So if you can review like structure of like IgM, TCR, your MHCs at least, and just kind of know them, it will help you out. Also know like when you're studying the immunoglobulins, like go over IgM, for example, is a pentamer, Ig alpha is a dimer, and all those things. Any questions so far? Yeah.
In your diagram inside, what does that stand for?
Oh, alpha 1. So alpha 1, alpha 2, and alpha 3. Yeah. I know my handwriting kind of sucks. So just, if you can't understand it, you can always just go back to the PowerPoints and they have pretty drawings there. Yeah, it's going to be much better. So the numbers, this is question 17. The maximum number of HLA genes, or MHC genes, that Professor Weigel could receive from his parents in the event that he is homozygous for MHC1 and heterozygous for MHC2 is going to be what? 12. What would you say that? You just remember it, okay. So if you're homozygous for MHC1, let's say, right? How many total MHCs could you have maximum? If you're homozygous, just for MHC1, you can have 3. And if you're homozygous for MHC2, you can also have 3 MHC2s for a total of 6, right? Now, if you're heterozygous for MHC1, then you have 6 MHC1s, 6 types of MHC1s, right? And if you're heterozygous for MHC2, then you'll have 6 types of MHC2. So then if you're heterozygous for both, you have a total of 12. If you're homozygous for both, you have a total of 6. Now the question is asking you're heterozygous for 1 and homozygous for 2. Sorry, homozygous for 1, heterozygous for 2. Thank you. Number wouldn't change, but so then the total that you could have is 9. Does it make sense? Yeah? Cool. So this is mostly a trivia question. I think he asked this in one of the tests, but 18 says a mutation in the MHC locus will affect the beta 2 microglobulin gene. And it is false. Can anybody tell me why it's false? Anyone? It's just, yeah, go ahead. Yeah, so the beta 2 microglobulin, although it's part of the structure, if you can see it here, it's not going to be part of the DNA. Beta 2 microglobulin is just a protein that we add to kind of stabilize the whole thing. Yeah. Sorry. False. So, MHC-TCR interaction for number 19. Let me just draw it really quick. So you have your, this is going to be your TCR. So this is your and then your MHC. Let's say it's going to be an MHC 1, right? Can you see this? There you go. So okay, so the all right, so this is your TCR up here, MHC1 down here. You can see that your TCR will have CDRs 1 through 3 on each chain, on the alpha and on the beta, right? And all you have to remember is that when the TCR and the MHC are interacting with each other, your CDRs 1 and 2 on each side, on beta and alpha, are kind of going to be on the sides, okay? So those two will interact with the MHC itself, right? Now, the CDR3 regions on both alpha and beta, those are going to be interacting with the peptide, which is this blob that I have in the middle, right? Will interact with the peptide. And if you think about it, it makes sense, because which one is going to be the most diverse CDR? CDR 3, right? Great. So the one that has the most diversity is going to interact with the thing that's most likely to be different every time, right? So, which is the peptide, whereas CDR 1 and 2 are going to be a little bit more conserved, and they'll always be able to interact with that MHC. Okay? Yeah? Cool. So then, to answer 19, it says, when interacting with a TCR, the peptide loaded on the MHC will mainly interact with the CDR3 region of the TCR, while the CDR1 and 2 regions will interact and recognize the MHC. Make sense? Yes? Okay, cool. So we're going to talk about SID now. Who can tell me what SID is really quick? Severe combined immunodeficiency. What's the reason for SID? RAG1 and 2 are not working, right? It can either be like one of them or both of them, but it's just RAG1 and 2 are not working. And that's not going to allow the person to do any somatic recombination on B and T cells, and you just kind of don't have an adaptive immune system, right? So which of the following is true about SID, or are true? 8 says that T cells are functional because of SID. Are the T cells functional? No. T cells are not functional because with SID, TCR recombination is affected, right? So this is true. One of the RAG genes is affected, which does not influence TCR recombination. Is this statement true? It's false, because one of the RAG genes is affected and RAG doesn't work, you're not going to be able to make a TCR, right? So the correct answer is going to be B. So this is kind of like a clinical case. It's saying that little Jimmy was taken to the doctor because of upper respiratory infections that he does not seem to be able to clear. His differential blood cell count shows that Jimmy has a normal lymphocyte count, but then when they do a genetic mapping of his HLA genes or MHC genes, they found out that his TAP proteins were affected, right? They had a mutation and they weren't working. So a mutation in your TAP genes is going to cause which disease? Someone? Bare lymphocyte, right? And what bare lymphocyte causes is you can't load any MHC1, right? Because the TAP proteins are the ones that will allow peptide to go from cytosol to your to the ER, which is where your MHC1 is loaded. So if you can't transport the peptide from one place to the other, you don't have any MHC1 loading, and that's bad. So then the correct answer is going to be E for that one. So HLA-A. What is HLA-A? It's a type of MHC, right? Is it going to be MHC1 or 2? How can we tell if it's, when we're talking about HLAs, if we're talking about MHC1 or 2? Anyone? Yeah, A, B, and C are going to be MHC1. And then the other is going to be, I think I heard it somewhere. No? DP, DQ, DR, remember? So those are going to be MHC2. So HLA-A will present antigens to CD4 or CD8 cells? Remember, it's MHC1. CD8. And it's one type of MHC1. You also have HLA-B and HLA-C. HLA-B here is misspelled. Oh well. So then the correct answer is going to be E. Cool. So your stages in B-cell development. Can somebody just sing them out loud? Early pro, late pro, large pre, small pre, immature, mature. Remember? Cool. So just as a quick review, in which stages do you have your heavy chain rearrangement? And so correct, and look at D, right? And tell me, between early pro and mature, where do you have your heavy chain rearrangement? Is it going to be at the beginning or at the end? Heavy chain. At the beginning, right? Now, the middle, remember, it's going to be your checkpoint. So in the checkpoint, you're not really going to have any rearrangement because you're just checking that the blank chain is working. I was about to give the answer away. And then in the small pre and mature, you continue doing the rearrangement, right? We said the one that you want to work on first is going to be out loud. Heavy chain, yes. So you work on the heavy chain during early and late pro. In large pre, you stop, make sure it's working. And then the small pre, you work on your light chain, right? And then your immature and mature cells, what's the difference? Which one's which? Sweet, awesome. So immature B cells, you can recognize them because they only have IgM on their surface. Mature B cells, you can recognize them because they have both IgM and IgD, right? Where do you find your surrogate light chain? In which stage? What's that? Large pre, awesome. Because that's where your checkpoint is, and that's where you use that surrogate light chain. When are RAG 1 and 2 going to be turned off? At the checkpoint, which is large pre, right? Did you have a question? V pre B and lambda 5. Those are the parts of your surrogate light chain. V pre B and lambda 5. What's up? What's that?
The checkpoint is reached with the entire stage of large pre, or is it a transition between large and small?
What do you mean? So that you're asking when the checkpoint is happening? your middle, right? So you start with, so to be more specific, early pro you have D to J recombination. Late pro you have V to DJ recombination. Your large pre, that's when you stop and you just check and make sure that your heavy chain is working. That's your checkpoint that if the heavy chain is working will allow for your B cell to continue and not die, right? Also remember, if the B cell messes up here in the heavy chain rearrangement, it has how many more shots to try for a new heavy chain? 1, right? Because you have another chromosome that, the homologous chromosome will carry the same genes, basically. Now, once everything's working well, then you move on to the small pre, where you do the light chain. There, you're going to have a total of 4 shots because you have, it can either be, what are the isotypes for the light chain? Kappa and lambda, right? So you have 1 kappa, 1 lambda on each chromosome, so you have 4 shots total, right? And then if all works well and you don't recognize self-antigens, then you can be a B cell, basically. Does it bring memories back, all this that I'm saying? Yes? Okay, cool. So 24. A large pre-B is distinguished by what? So large pre-B is where we have the checkpoint, right? So large pre-B is going to have the surrogate light chain. So your pre-B cell receptor consists of a heavy chain and a surrogate light chain, which is with its Ig alpha and beta on the side. And then the surrogate light chain will test whether the heavy chain will be able to bind to the light chain, basically, right? Cool. So correct one is going to be E. Now going back to that RNA splicing again. So where's the drawing? I lost it. There it is. Going back to this once again. A mature B cell will choose whether to produce IgM or IgD through RNA splicing, right? Because remember that the first antibody you ever produce is going to be IgM, right? But when the B cell is maturing, it's going to need both IgM and D on the surface when it's a mature one. So instead of having to make all the IgM first and then cut the DNA and make the IgD, you can just leave both genes, transcribe it to RNA, and then just choose whatever you need and leave it on, and whatever you don't need, you just cut it out, right? Now, once you switch to IgG, for example, that's when you go back to the DNA and you cut out the M and D and you leave the G. So you won't be able to go back to whatever isotype you had before. Bless you. Does it make sense? So with IgM and D, you can always switch back and forth through RNA splicing. Once you switch to G, A, or E, that's when you can't go back to M or D, or technically any of the others, right? You can only keep whatever is downstream of that gene. But just know that that switch between M and D can happen all the time, but then once you switch to G or E or A, that's when you can't go back to M and D. Okay? Yeah? Cool. So the correct answer here is going to be D. A mature B cell will choose whether to produce IgM or D through mRNA splicing. So a little bit more about your B cell maturation. The pre-BCR is also known as the blank checkpoint, which is a blank signal that blanks apoptosis. So it's going to be your first checkpoint, which is going to be a positive signal that will prevent apoptosis. We just talked about this. You remember? Yeah? Okay. Cool. Translocation of the MYC gene. Will it give you superpowers? No, it'd be cool, but no, it does give you lymphoma though. So is it lymphoma? Yeah. Burkitt's lymphoma. Just a tip. As you're studying, just write down all the diseases that you've seen throughout the semester. It's not that many. I'm sure it's less than 10. Like I'm 85% confident about my 70% confidence that's not that many. But no, yeah, it really isn't. You don't have that many diseases, so just, write them down on the side, make some flashcards, and make sure you know them, okay? That's why I'm just putting them in here, like in random places, just to make sure that I spur your memory and you remember it, hopefully, for the test. More B cells. This is talking about B1 cells. So B1 cells are a little bit different than normal B cells, B2 cells, because why? Anyone? What's different about, what's the difference between a B1 cell and a B2 cell? It doesn't need a T cell, right? It's going to use your thymus independent antigens to get activated, but it won't be able to do any isotype switching once it gets activated. It's just going to be able to secrete IgM. And those are the B cells that are present when you're a fetus, basically, right? So when you're in there, those are the ones that are going to help you if you were ever to encounter an infection while you're in mom's belly, basically. And they're special because they don't have as much diversity as normal B cells do, and that's because they don't have TDT. Just letting you know. I'm just going to say it. They're going to join their VD and J sequences in a different way, and they're not going to have that TDT that just adds random nucleotides in between, so that's why they're just less diverse, basically. Plasma cells. So you can get any type of B cell and turn it into a plasma cell, true or false? Mature B cell, right? Because a memory cell can get activated and can turn right away into a plasma cell, right? A somatically hypermutated centrocyte can get out of the follicle, interact with the T cell, and be told to turn into a plasma cell right away, right? And isotype switched B cells as well, the same thing. And then an activated B cell that just came into the lymph node for the first time and just gets activated also can turn into a plasma cell, right? Do you all remember that? Cool. So all of the above. So T cell maturation. So let's go over T cell maturation really quick. You begin in the bone marrow, a stem cell migrates to the thymus, right? In there, the stem cell is not committed to become a T cell yet, right? But it will receive that Notch1 signaling along with IL-7, and it's going to make some changes that will commit that stem cell to turn into a T cell eventually, right? That's when we call it a committed progenitor cell. Once that happens, your thymocyte, which is, we call it thymocyte at this point, is going to start doing somatic recombination, right? It's going to work on its beta chain first. It's going to stop, do that checkpoint, make sure that the beta chain is working right. Then it's going to start again, work on the alpha chain, right? And once it finishes the alpha chain, it needs to do two more things. T cells need to be able to recognize self-MHCs, right? And then they need to be able to not recognize self-peptides, right? So positive and negative selection. I see it as, think of the thymocyte as a student at UCF that needs to fulfill prereqs. But in this case, if you don't fulfill the prereqs, you kind of die. We just, take the class again and hope for the best. So going back to the whole somatic recombination, in the checkpoint in between alpha and beta chain recombination, You're going to have that pre-T cell receptor form to make sure that the beta chain is working. When the beta chain shows that it's working properly, you're going to have a couple of signals originated. So it's on the slides, but the first one is to stop beta chain rearrangement. The second one is it's going to stimulate proliferation, right? So because everything is going well so far, that thymocyte is just going to divide a bunch before it keeps going. Then it's going to express CD4 and CD8. So it's going to turn from double negative to double positive thymocyte. And then the fourth one is to become permissive to start that alpha chain rearrangement. Okay? So here it's asking which of the following is not a signal, right? And the one that's not a signal is going to be B, because it says that it stimulates expression of either CD4 or CD8. It's going to stimulate the expression of both, so it becomes double positive. Then, when the alpha chain is ready, and you have TCR good to go, you go through positive selection, where you recognize your MHC, right? When it recognizes the MHC, that's when your T cell, your thymocyte is going to turn into a single positive cell, because if it recognizes an MHC1, it's going to turn into a CD8 cell, so it's going to get rid of the CD4 on its surface. If it recognizes an MHC2, it's going to keep its CD4 and get rid of the CD8. Does it make sense? And then the next step is the negative selection, where you recognize self-antigens. And as long as you don't recognize any self-antigens, then you're good to go. During that whole process, we get rid of like 98% of the cells that are in there, because it's very selective, basically. Okay? So once the first chain of the beta, So once the beta chain of the TCR is rearranged and the cell is finished proliferating, the RAG genes are turned back on. Rearrangement of the alpha chain locus is induced. There is a bigger chance for that rearranging thymocyte will become gamma delta than alpha beta. A and B, all of the above. So put it this way. Alpha and beta, A and B are correct, right? Once you make sure that your beta chain is working, you turn RAG 1 and 2 on, and you're going to start working on your alpha chain, right? But C is not correct, because there is not a bigger chance for the T cell to become gamma delta. There's a bigger chance for it to become alpha beta. Remember, just like B cells, we have B1s and B2s. With T cells, we have alpha beta gamma delta T cells. Gamma delta T cells are just the weird ones that we don't really care about because of reasons. We just don't study them. Just remember that alpha beta T cells, they're going to need to go through the whole maturation process in your thymus. Your gamma delta T cells, if they choose to become gamma delta in the somatic recombination process, they're just going to finish working on their genes and then get out of there. They're not going to go through positive or negative selection, just because gamma delta T cells are not they're not MHC dependent. They can bind to antigen that's just floating around. Question?
So the option left B is not the most set option, duration of MC.
Oh yeah, that's a good point. So if you notice, it says D, alpha beta T cell. That's like a continuation of C, but for some reason it thought it was a different line. So the correct one is going to be this one, alpha and beta. All right? Make sense? Okay. Any questions so far? No? Okay. So the pre-T cell receptor is composed of a fully formed TCR? No. Is it going to be a beta chain and a pre-T alpha? Is it going to have CD3 and zeta chains? Yes, always, right? That's the safest bet is that CD3 and zeta chains are always going to be present when you talk about TCR. So then the correct one is going to be E, options B and C. So the rearrangement of the alpha chain locus continues throughout the process of positive selection and stops whenever the T cell is positively selected. So positive selection is when your MHC, when your thymocyte is recognizing your MHC, right? And if your thymocyte is having a hard time to recognize anything, right, it's going to start freaking out because it's going to be like, yo, I'm not recognizing anything, you know, I'm going to be induced to die by apoptosis, I don't want to die, you know, I have plans. So what it does, it's going to continue working on that alpha chain, make a couple of mutations here and there, try to pick a different V and J segments, and when it can make something that actually works, that actually recognizes an MHC, it can continue. Now, if it doesn't recognize anything at all, eventually they'll die by apoptosis. Does it make sense? Yeah? Okay. Because remember, with T cells, for the alpha chain, you have multiple attempts for rearrangement. It's not like with B cells that with B cells in your heavy chain you have 2, and your light chain you have 4. Okay? Cool. Tregs. Oh, that's crooked. Sorry for those of you who have OCD. That was crooked. Anyhow, what's important about Tregs? They have FOXP3, awesome. And it's a transcriptional repressor, transcription factor. What does FOXP3 do for that Treg? We didn't really talk about it, but just any guesses? I don't know who talked. Back there? Oh. Yeah, so transcription factors are proteins that will allow you to transcribe genes. So what it will do, it will allow that T cell to, you know, use certain genes to be able to regulate other T cells. And in general, your immune system, right? That's why Tregs are super important. And that's why that FOXP3, you're only going to see it in Tregs, I think. There's very few cells that can use FOXP3. Yeah. Now, is it a type of CD4 cell? Yeah, right? The other types of CD4 cells are going to be Th1, Th2, Th17, T follicular helper, and then the Tregs. Cool. So this question is asking about which regions of your CDRs you're testing during positive selection. So in positive selection, your TCR is recognizing what? What are we recognizing in positive selection? MHC, right? It can be 1 or 2, doesn't matter, because we just want one type to be recognized. So we talked about this drawing. Remember, CDR1 and 2 will interact with the MHC, and CDR3s are going to be interacting with the peptide, right? So in positive selection, we were trying to recognize MHC. We only care about CDRs 1 and 2, right? So then the correct answer is going to be C. Positive selection tests whether the CDR1 and CDR2 regions of the TCR can bind to an MHC. Okay? Cool. What's that? Yeah, so the answer is going to be C. CDR1 and CDR2 are what you test to make sure that you can bind to the MHC. Question 36. When the T cell is screening the peptide MHC complex on the APC. So here we're talking about T cell activation. We're jumping a little bit forward to chapter 7, I want to say, I think. But it's the T cell mediated immunity chapter. Remember that we have a macrophage that's just chilling in one of the tissues, right? And let's say I fall, scrape my knee, and some bacteria gets in my knee, right? I'm going to have some dendritic cells there that are going to engulf that bacteria, process it, present it on an MHC class 2, and travel to the nearest lymph node, right? Remember that in the process, your dendritic cell is going to grab its PRRs, pattern recognition receptors, so like the Toll-like receptors, mannose receptors, and all those things. Those are also going to get activated. So then the dendritic cell is going to be able to produce B7 and also put that out on the surface, right? So once the dendritic cell gets to the lymph node, your lymph node is going to be full of CD4 cells that are going to see the dendritic cell. That are going to be like, oh, let's see what, Mr. Dendritic Cell brought it to us today, basically. And they're going to start scanning around and see if there's any recognition. If the T cell can recognize the peptide MHC complex, that's when you get an immune response, right? So before all of that happens, you need to have interaction between a couple of things. So MHC So this is the dendritic cell. You're going to have your MHC here. Which kind of MHC is it going to be if it presents to CD4 cell? MHC class 2. Alpha 1, beta 1, beta 2, alpha 2. Okay? You're going to have B7. I do not remember the shape of the protein in the PowerPoint, so let's just pretend that this is B7, right? And then you're going to have adhesion molecules as well. I'm just going to make a ICAM 1 here. Let's say, yeah, an ICAM and a DC sign. DC sign is an adhesion molecule that's only on dendritic cells, just so you guys know. Now, the T cell, might have to zoom out a little. Yeah, there you go. So your T cell is going to use its TCR to recognize the peptide, right? It's going to have your CD4 that it's kind of interacts with your MHC over here. It's going to have, what interacts with B7? CD28 at the beginning, then CTLA4. This is at the beginning, so it's going to be CD28. CD28. Oh my God. I'm not used to talking so much. And then more ICAMs and LFAs that will bind to ICAMs. So the adhesion molecules will interact with each other, right? Remember, T cell, this is a TCR. All right? So remember the interactions that happen between proteins on one cell and then on the other, okay? What happens as a result of this? Yeah, you get T cell activation. This is where the whole ZAP70 activating phospholipase gamma, PLC gamma that cleaves PIP2 into IP3 and DAG and all that, that's when it happens, right after this. right? Before the T cell gets activated, your T cell will start making IL-2, which will promote replication, basically. Your T cell is going to divide a lot just because it's the one that can recognize the peptide. And then once it divided and, once you have enough clones, that's when you switch the CD28, you switch it to a CTLA4. All right? You switch it to a CTLA4 and you stop all those rounds of division, and your T cell now, it's ready to differentiate into Th1, Th2, Th17, T follicular helper, or Treg, depending on which cytokines are there in the environment. Does that make sense? Yes? Any questions so far? What's up? We don't really talk about what happens that makes it switch. It's just once you have enough clones of that activated T cell, that's when you switch. And CTLA4 binds to B7 as well. So then because CTLA4 has a higher affinity to B7 than CD28, that's when the whole system is repressed and your division stops, and then that's when you really get the activation because your T cells are going to be the ones that they're going to differentiate, basically. Make sense? Kind of? What did I not answer?
So it just switches
Because
because that's what's going to bind better?
that's going to what?
That's what's going to bind better with the B7?
It's not because that's what's going to bind better. It's because it divided. You have enough clones of the T cell that you don't need to continue divisions. You don't need to make any new T cells. Because if you have what results from this is a loop that keeps enhancing itself, right? Because you keep making IL-2 that will tell the T cell to continue to divide, right? And it's just going to keep going and going and going. So if you don't stop that, what do you get? Cancer, yeah. So that's when you have your CTLA4 to just regulate, right? And make it stop whenever you need to, basically. So going back to the question, when the T cell screening for the MHC peptide complex on the APC, which stands for antigen presenting cell, in this case it's going to be the dendritic cell, but APCs are also macrophages and B cells, remember? The T cells LFA1 binds to ICAM 1 or 2 on the APC. ICAM 3 on the dendritic cell binds to DC sign on the activated T cell. SP1 receptors will be suppressed if the T cell is able to bind successfully to the peptide complex. So which one of these is false? There's one statement that's false. B, right? Because DC sign is going to be on the dendritic cell, always. So if this was backwards, then the correct answer would be all of the above. But the correct answer is A and C. The SP1 receptors, just remember, I think it's like sphingosine 1-phosphate receptors. They're just receptors that will tell your T cell to leave the lymph node if they've been there for long enough that they didn't recognize anything. So then they can just go into circulation again and go inside another lymph node and try their luck in there and see if they can recognize something. So which of the following statements about co-stimulatory protein B7 is or are true? B7 ligand CTLA4 has 20-fold binding strength less than CD28. False. Awesome. That's because it's more, not less. B7 on the surface of the T cell binds to CD28 on the surface of the APC and is a signal that indicates whether the peptide loaded on the MHC2 is exogenous. This could be true if it said that the B7 is on the dendritic cell and CD28 on the PAPC, right? So this is also false. So then the last one says that when CTLA4 binds to B7, the T cell proliferation slows down. That's correct, right? Yeah? Cool. A T cell that recognizes a self-antigen will most likely activate and proliferate, right? If a T cell recognizes self-antigen, will it get activated and proliferate? No, because you get a autoimmune disease. So in an ideal world, it won't happen, but sometimes it happens. But for test purposes, you know, a T cell that recognizes self-antigen does not get activated. Will be rendered anergic because of the presence of B7 on the APC. This is also wrong because it will be rendered anergic because of the lack of B7. Okay? It will not be rendered anergic because of the presence 38E. So disregard C because the way I worded this, it just makes it weird. So none of the above. Just know that if a dendritic cell were to load to eat something that's from your own body, let's say a dead epithelial cell that was just floating around, the dendritic cell can still go to the lymph node and present it to T cells. But it's not going to have B7 on the surface because it's not something that's exogenous. It's not something that comes from the outside, right? The dendritic cell realizes that it's part of us. So it's not going to put B7 on the surface. So if a T cell approaches the dendritic cell and recognizes that epithelial cell, because you don't have any B7 on the surface, your T cell won't get activated and it's just going to be rendered anergic, and then eventually it's just going to die. Okay? That's all you need to know for this one. Cool? All right. All leukocytes are lymphocytes. I heard false and true. All leukocytes are lymphocytes. Are you sure? Okay. Because remember, what falls under the lymphocyte category? TB and NK. What falls under the the lymphocytes, TB and NK. What falls under the leukocyte category? Everything, right? Any white blood cell. So all lymphocytes plus Ben, macrophages, dendritic cells, monocytes. All right? When you get this question, make sure you don't screw it up, okay? It's like an easy 1 point. Cytokine differentiation. Now, this is a little specific. I don't know if you will need to know this, but I still put it on. IL-4 will cause differentiation of an activated T cell into which one of these? Th2, yeah. This is from the one chart in your T cell mediated immunity chapter that has all the different types of T cells and then all the cytokines. You didn't need to study that for that one test, but I don't know if, what's that? Yeah, I know. I don't know if you'll need to know it for the final. If you know it, then, you know, good for you because it's, you know, more things that you know for the test. What's up? Yeah, but we're talking about T cells here. But yeah, he's pointing out that IL-4 and B cells will induce an activated B cell to turn into a memory B cell. In that same regard, IL-10 will induce the B cell, the activated B cell, to turn into a plasma cell. Yeah, good point. Thank you. Leprosy. Do you all love leprosy? Do you remember leprosy? Yes? No? So if you had to choose to get one of the two, lepromatis or tuberculoid, which one do you choose? What's that? She's saying tuberculoid. Do you agree with her? Or would you rather have the lepromatis? What? I agree with her. Yeah. So the difference is what kind of T cells you get activated with the disease. With tuberculoid, so leprosy is a bacteria that infects your macrophages. It goes into their vesicles and prevents them to join their lysosome with their phagosome. So whenever a macrophage eats the bacteria, it will never be able to join the phagosome and lysosome and destroy the bacteria, right? So people have bias toward either a Th1 or a Th2 mediated immunity. Now, who can tell me what a Th2 response does? If you have Th2, what are you going to have as a result? Yeah, B cell activation in general, right? So Th2 will cause B cell activation. So if you have a bias, again, if you have a bias for Th2 differentiation or activation, and you have the lepromatous leprosy, then you're going to get B cells activated, and you're going to make a bunch of antibodies. Do you think the antibodies are going to do anything against the bacteria that's inside a macrophage? No, right? Because the antibodies are not going to be able to like drill a hole into the macrophage and kill the bacteria. Whereas if you have tuberculoid leprosy, then you'll have a bias towards Th1 cells, and Th1 cells do what? Th1 T cells. What's that? Activation of macrophages, right? So with a Th1 bias with the tuberculoid leprosy, you'll actually basically get activation of more macrophages, so you'll get a higher chance of just clearing the infection, right? With those macrophages that haven't been infected yet. So your lepromatous leprosy has a high infectivity because, and it causes a differentiation and bias of Th2 cells. And we'll also have a higher concentration of IL-4, 5, and 10. So then the correct one is going to be B and C. Any questions so far? Yeah, what's up? Why do we have what? That's just because of the activation of those cells. Yeah. This is just one of the other diseases that you have that it'd be wise if you wrote them on the side and just wrote down the main points on it. Okay? Any questions about this? No? All right. Activation of macrophages. So with macrophage, remember, you always have a primary and a secondary signal. Primary signal is going to be your interferon gamma. Secondary signal is going to be your CD40 ligand. Okay? Who's going to secrete interferon gamma? I don't know what you said. Okay. Who secretes interferon? NK cells also. Remember, right? So and remember that macrophages, when they get activated, they secrete IL-12, which also activates NK cells, right? So it's like a so the first signal of macrophages that is needed for macrophage activation is receiving interferon gamma. Interferon gamma is produced by NK cells. And remember then that when you have inflammation produced by a macrophage, you have a bunch of cytokines produced, right? Who can tell me those cytokines? TNF-alpha, IL-6, IL-12, CXCL8, and IL-1, right? IL-12, produced by the macrophage, recruits NK cells, which will make interferon gamma, which will activate macrophages, right? So it's just a big circle of life that will allow you to fight infection better, right? And then once you clear the infection, you're going to have other mechanisms that come in and just kind of stop everything and slow everything down. One of them being the Tregs, for example. So your primary signal is going to be your interferon gamma. Your secondary signal is going to be your CD40 ligand. When an activated naive B cell starts interacting with a T follicular helper, remember that an activated naive B cell has never been activated before. This is the first time it's getting activated. So it's going to secrete what first? IgM, right? And it's also going to be told to, do a bunch of divisions, cycles, replication. And it can either, the clones of the B cell will either be able to turn into plasma cells or go into the follicle and start somatic hypermutation and isotype switching. At the same time, your T cell, in that occasion, it will secrete IL-5 and 6. Let's see. 43. So the T cell will release IL-5 and 6 into the medullary cord. That's going to allow your B cells to just receive those signals that they'll need to start doing the different proliferation. IgM will be the most abundant antibody produced because it's the first one, right? And it's a naive, activated naive B cell. And then the B cell will form the conjugate pair with the T cell, right? And that conjugate pair will allow CD40 ligand interaction. Now, who can tell me what CD40 ligand did on the B cell? Because we just saw it on the macrophage. Yeah, so you have activation in NF-kappa B, which will allow for ICAM production. ICAM is an adhesion molecule, and it will allow to produce that really tight binding so that the B and T cell can kind of hang out together for a couple of days and, mediate the immune response. The CD40 ligand comes into play when the B cell comes back from somatic hypermutation and isotype switching, because that's how the B cell will also get the survival signal. Do you all remember what the survival signal is for the B cell? You have the letters but scrambled. BCL-XL. Yeah, it's BCL-XL. So what basically happens is a B cell has an antigen from follicular dendritic cell, puts it in, takes it in, puts it out on an MHC class 2, goes to the follicular helper cell on the border between the B and T cell area. When a T follicular helper cell recognizes whatever the B cell is carrying, that's when that conjugate pair is going to be formed, right? CD40 ligand interaction. You're going to have a lot of division so that some of those new divided B cells can turn into plasma cells, and the rest can go into the follicle and do some somatic hypermutation and isotype switching. When the 2 or 3 B cells that were able to get out of the follicle with a better BCR come back to the T cell, they're going to interact again, their MHC2 with the TCR, so that you have CD40 ligand interaction, and that's when the BCL-XL is expressed in the B cell. The B cell is allowed to live, and then the T cell is going to tell the B cell either turn into a plasma cell or turn into a memory cell, depending on where you are in the infection. Make sense? That's like chapter 8 in a nutshell, pretty much. But it's like a back and forth, in and out of the follicle, basically. Cool. So this is all of the above. Which of the following statements regarding a recently somatically hypermutated and isotype switched B cell is or are true? It will have to compete to interact with a T follicular helper, which is true, right? Because you have a lot of clones of B cells that they're all trying to improve their BCRs, and they have to show it to the 1 or 2 T cells that are there available. So this is true. They will die by apoptosis if the B cell is induced to produce the BCL-XL. That's not true. And can turn into a plasma cell or a memory cell without the signals from the T follicular helper. Is that true? No, that's also false. With B cell activation, any step of the B cell, anything that the B cell does, it needs approval from the T cell. It's like an annoying little kid, right? That's always looking at you to see if he or she is doing the right thing. Pretty much, yeah. So for somatic hypermutation, it needs the T cell to tell it to go into somatic hypermutation and isotype switching. For turning into a memory cell, it will need the T cell. And for turning into a plasma cell, it will also need the T cell. Cool. So when an infection just started, right, we have an increasing amount of pathogens in our body. Your T follicular helpers are expected to secrete more blank to induce B cells to differentiate into blank. Yeah, so if your infection is starting or worsening, you want to make more antibodies, so you need more plasma cells. B cells turn into plasma cells when IL-10 levels are high, right? So correct one is going to be C. So complement again. Now we're going back to like the first chapter, and we're talking about regulation of complement. Which of the following are used to slow down complement in your body, basically? Is it factor P, DAF, MCP, both DAF and MCP, or none of them? What's that? You said E or D? D, yeah. So remember, factor P is going to be the accelerator. It's going to stabilize your C3b big B little b, your C3 convertase, so that it can make more complement. It can cleave more complement. I'll get to you in a second. Factor H and I are going to stop complement in the surface of a bacteria. H and I. H and I. Yeah. Factor H and I will slow down complement on the surface of a bacteria. And then DAF and MCP, they're going to slow down complement on the surface of your own cells, right? Because complement can get activated in your own cells. So we all have DAF and MCP to avoid that happening. Okay? Question? Oh, I don't remember. I don't know. Yeah. I'm sorry. Factor P is the accelerator. It stabilizes your C3 convertase so that it can cleave C3 faster. All right. Oh, man. I'm sorry about this. I hate when this happens. So, which of the following scenarios is considered a passive form of immunization? A form of passive immunization. IgA crossing the placenta to enter the fetus of the bloodstream. Is that passive immunization? If it was IgG, it would be. But IgA does not cross the placenta, remember. Oh. Does it make sense? Yeah? So because you have IgA here, that's wrong. Somatically hypermutated and isotype switched IgG that are being released by a plasma cell in a secondary lymphoid tissue during an infection. Is that passive immunity? No. That's active immunity because you're making it yourself. Steve Irwin getting antivenom after being bit by a black cobra because he felt like petting the thing. That is, yes. Great. Awesome. So 48 is talking about chapter 10. So that's the whole GALT chapter. And it's basically asking which gradients do B cells follow when getting into a lymph node in the MALT, right? So remember, all naive B cells can follow a CCL19 and 21 gradient to go through the HEV, right? But then once a B cell gets activated in the MALT, it needs to be exclusive, right, for the MALT. It cannot be activated again if it goes into circulation and it ends up being in like a lymph node in my armpit. It can't get in there, right, because it belongs to the MALT. So what it does, it's going to get rid of the receptors for CCL19 and CCL21, right? And it's going to start recognizing CCL25. At the same time, it's not going to use selectins anymore, but it's going to use another adhesion molecule called alpha 4 beta 7. And this is actually wrong here. It's a typo. This is supposed to be alpha 4 beta 7, not alpha 1. So naive lymphocytes can circulate through the lymph node, spleen, or any secondary lymphoid tissue because they follow a CCL19 and 21 gradient when entering the tissue through the HEV. But once a lymphocyte has been activated in a MALT lymphoid tissue, it will follow a CCL25 gradient and will express the adhesion molecule alpha 4 beta 7. Make sense? It's just a typo. Yeah, I just said, make sure you correct it. Alpha 4 beta 7, not alpha 1 beta 7. Make sense? Yeah? I'm just going to... So 49 is B, 50 is D. I only have 5 minutes, so I wanted to see if anybody has any questions. 49 is B and 50 is D. I just wanted to skip forward really quick and see if anybody had any questions so far.
What do you think is the most important knowing besides the diseases? You mentioned diseases.
Yeah, so he's asking about what do you think it's the most important thing to know or things. As you're reviewing stuff, make sure you know all those things that keep showing up over and over and over, right? So we see B cells over and over, so I definitely know B cells in general, what they do, and T cells as well. If you can, now that you've finished studying for the whole book basically, I try to see if you can make yourself a scenario, right? From a B cell in the bone marrow to a B cell activation, from a T cell in the thymus to T cell activation. See if you can make yourself a scenario with an infection in that it's dealt with your innate immune system, right? With your macrophages, neutrophils, and so on and so forth. Diseases, like I said, are going to be important. And what's that? Yeah, definitely. So as you're reading through the PowerPoint, I hope that you've noticed that he has certain things that are written in red, right? So as you're scrolling through, make sure you know and you understand those things, okay? Before we move on, does anybody not have the evaluation? There you go. So if you can fill those out in the meantime, but make sure you know all those things as well. Complement, antibodies for sure. Yeah, no problem. Go over the my practice tests if you want to give them a try. They're all in the web, not web courses.
Is it possible if we can go to the very first page?
Yeah. Switch. There you go. As you're filling that out, make sure you bubble in 37, please. I'll post it now. Yeah, I'm going to go to the office. What's that? Oh, they should be there. Yeah. What else? Yeah, I mean, I think that's it. Oh, before I let you go, make sure you sleep, guys. For real, right? So, pace yourself as you're studying, but then the best thing you can do is just take a good nap. Take a solid 8 hours of sleep. That's gonna relax you. It's going to reset your brain so that when you get there, you can just take the test and it's going to feel like a breeze. Before you sleep, maybe, if you have a bathtub, just, turn on the hot water and just relax for a couple of minutes or something. But, make sure you sleep, please. Sleep still, yeah.